When you're ready.
Hello everybody, my name is Elizabeth Long and I am here today to talk to you about my AP research project. The specific question that I have been studying and attempting to answer is what is the optimal dosage of tryptophan to administer to tetrahymena thermophila to see adequate behavioral changes. Alright, so here are the various parts of my research that I will be addressing today. I will start by talking about how I arrived at this specific question, then I will explain the specific goals I have been trying to reach, then I will describe my methodology, results, conclusions, and implications. So first I'd like to talk to you about how I generally arrived at the question I have been studying. So going into AP research at the start of the school year, I knew right away that I did not want to experiment on human subjects. After listening to many people from previous years talk about how testing on humans can be unreliable, I decided immediately that I was going to test on the organism Tetrahymena thermophila. In AP seminar of my junior year, I was introduced to this organism. Tetrahymena thermophila is a unicellular eukaryotic organism, specifically a ciliate protozoan. It's commonly studied in biological research as a model organism due to its relatively simple structure, fast growth rate, and well-understood genetics. Scientists use tetrahymena as a model organism to study various biological processes, including cell division, gene expression, genetic recombination, and the effects of environmental factors on living organisms. Its relatively simple genome and ease of cultivation make it valuable for experimental purposes. Once I decided that I was going to be testing on tetrahymena, I needed to narrow down my focus and choose a specific topic. This led me to exploring one of the most fascinating areas of science, in my opinion, which is psychology and neuroscience. In my junior year, I started self-studying AP psychology because my school does not offer this class. Immediately, I was intrigued. The units on the brain, cognitive processes, and behavior sparked my curiosity, leading me to merge these interests with my research using tetrahymena. Understanding the connection between behavior and underlying neural processes became the cornerstone of my investigation. By incorporating principles from AP psychology and neuroscience into my study, I aim to bridge the gap between microscopic observations of tetrahymena and broader insights into behavioral changes influenced by external factors. Finally, I was getting closer to arriving at a final topic to study. However, I hit a roadblock and could not think of a specific area that connected psychology and neuroscience to tetrahymena. I had to think outside of the box, and I started thinking about areas in my own life that could be applicable to the topics I had already formed. The first thing that came to mind was nutritional supplements. From a young age, I've incorporated various supplements into my daily routine to address specific health concerns, ranging from digestive issues and allergies to managing anxiety and ADHD. This not only sparked my interest, but also presented a unique opportunity to explore their potential impact on tetrahymena, given their relevance to neurological and behavioral functions. However, I encountered a challenge in deciding which specific supplements to investigate, considering that many have already undergone extensive study and analysis. After much research, I stumbled upon the supplement tryptophan, which has room for further research. Tryptophan is essential amino acid, meaning that the body cannot produce it on its own and must obtain it through diet or supplementation. It plays a crucial role in various psychological processes, particularly in the synthesis of serotonin and melatonin, two neurotransmitters involved in mood regulation and sleep-wake cycles. In the context of research on tetrahymena, investigating the impact of tryptophan dosage on behavioral changes offers insight into its neurological mechanisms and potential applications across human and microbial systems. A study by Abdullah Badaway, published in 2013, focused on tryptophan's role as a precursor to serotonin and its impact on mood and cognitive performance in healthy individuals. The findings highlighted tryptophan's potential to enhance mood, specifically feelings of happiness and calmness, without negative cognitive effects. This study underscores the importance of dosage optimization, as participants experienced mood improvements with no adverse cognitive outcomes. Understanding the implications of tryptophan dosage on behavior not only contributes to our knowledge of neurotransmitter pathways and mood regulation, but also holds relevance for broader applications in behavioral research and therapeutic interventions. By building upon the insights from Badaway's study and investigating tryptophan's effects in a microbiome model like tetrahymena, we can unravel the nuances of dosage-response relationships, inform personalized supplementation strategies, and advance our understanding of neurobehavioral interactions. This research has implications for human health, personalized medicine, and the fundamental understanding of biological processes. To conduct my research, I began converting powdered tryptophan into liquid form for easier administration to tetrahymena. I diluted 1.875 grams of tryptophan in 250 milliliters of distilled water in a 500 milliliter beaker, taking approximately 10 minutes to ensure complete dilution. This tryptophan solution was then refrigerated for cooling. Next, I cultured the first set of tetrahymena in 10 labeled test tubes, allowing them to incubate for several days. After incubation, I examined a droplet under a microscope, recording observations and capturing media for comparison. Subsequently, I administered 1,000 microliters of tryptophan solution into each test tube and observed the cultures after 24 hours under the microscope, documenting any changes and comparing them to control cultures without tryptophan. I repeated this process in 3 trials. In trial 1, I increased tryptophan dosage until the tetrahymena ceased moving or showed significant behavioral changes. In trial 2, I aimed to find the optimal dosage by observing behavioral changes without reaching lethality. In trial 3 involved administering tryptophan in smaller increments to explore nuanced effects on behavior. Okay, so because it would take a long time to show and explain the results of each of the 30 cultures that I experimented on, I'm just going to show the ones with the most significant changes. In trial 1, after administering 1,000 microliters of tryptophan to the tetrahymena, nothing changed and they looked relatively the same. So I decided to give them an additional 1,000 microliters of tryptophan. After 24 hours, there was a significant more amount of tetrahymena present, and they were moving a lot faster when they had received no tryptophan. After I gave them an additional 1,000 microliters to make the total 3,000 microliters, the tetrahymena were continuing to multiply, but they were moving very slowly, and some were not moving at all. The test tubes were covered with mold on the top and bottom of the culture. In trial 2, I repeated the exact same process, except the maximum amount of tryptophan that I administered to these cultures was 2,000 microliters. The original cultures already had less tetrahymena than the ones on trial 1, so I was already experimenting on different grounds. All of the tetrahymena were moving very slowly, and some of them were not moving at all. After they received 1,000 microliters of tryptophan, they multiplied, and there was a lot more tetrahymena present than the previous culture. They seemed to be moving faster than they were before, but were still moving slower than the cultures in trial 1. After they received 2,000 microliters of tryptophan, they yet again multiplied in size, and they were moving at a much faster pace. This seemed like the appropriate amount of tryptophan to administer to them to see adequate behavioral changes, and they went from looking depressed to lively and happy. In trial 3, I repeated the same process except with smaller increments. I started with 20 microliters of tryptophan immediately after I examined them under the microscope for the first time. However, I knew that 20 microliters would show no improvements whatsoever, so I did not look at them under the microscope after this increment and instead administered the additional 1,000 microliters right away, except in 2 500 microliter increments. After examining these cultures under the microscope 24 hours later, the results were shocking. The tetrahymena had decreased in size and were showing impairments in their ability to swim. They were moving a lot slower than they had before, and many of them were already dead. This was shocking to me because in the trials before this, 1,000 microliters of tryptophan had shown no improvement, and 2,000 microliters showed improvements in their speed, numbers, and size. I thought that 1,020 microliters would be the happy medium, but it turned out to be the opposite. So what do these results tell us? First and foremost, we've seen that the effects of tryptophan on tetrahymena behavior are highly dependent on the dosage administered. Lower doses didn't seem to produce significant changes, while higher doses had varying effects, from enhancing movement to potentially impairing behavior. Interestingly, my second trial showed that administering 2,000 microliters of tryptophan seemed to hit the sweet spot. I saw increased tetrahymena numbers, improved movement, and overall better behavior without reaching a point of lethality. This suggests there's an optimal range for observing the desired changes. On the flip side, my exploration with smaller tryptophan increments in trial 3 revealed some unexpected responses. Contrary to what I thought, smaller doses didn't always lead to better behavior. In fact, in some cases, I saw impaired movement and higher mortality rates. What I've learned here is the importance of keen observation and meticulous recording of behavioral changes. By closely monitoring tetrahymena thermophila's response under the microscope, I was able to detect subtle shifts and gain valuable insights into how tryptophan affects their behavior. Looking ahead, these findings open up avenues for further research. I could explore the underlying mechanisms behind tryptophan's effects, explore its long-term impact, and consider other factors that might influence the response to tryptophan. As we look ahead, the implications of my findings extend far beyond the confines of the classroom. My research explores the intricate workings of neurotransmitter pathways and behavioral responses, offering insights applicable to a wide array of organisms, including humans. This paves the way for deeper investigations into neurological processes and behavior. The optimal dosage range I've identified holds promise for personalized medicine. Tailoring tryptophan supplementation based on individual responses could revolutionize treatments for mood disorders and other health conditions, promoting better outcomes for patients. My research serves as a valuable education tool, showcasing the scientific process from hypothesis formulation to experimentation and data analysis. It provides a hands-on learning experience for students interested in biology, neuroscience, and experimental research. Looking ahead, further research could explore additional factors influencing tryptophan's effects, such as environmental variables and genetic diversity. Unraveling the molecular mechanisms underlying tryptophan-induced behavioral changes open doors to deeper scientific inquiries and potential therapeutic developments. In essence, this study not only advances scientific knowledge, but also holds promise for improving personalized health interventions, fostering environmental awareness, and inspiring future generations of scientists.
Okay, thank you. Excuse me. How did the choices you made when designing or implementing your research method impact your research process?
Could you repeat the question?
Sure. How did the choices that you made when designing or implementing your research method impact your research process?
I think that definitely doing 3 trials instead of just 1 showed an overall varying of my results, which kind of concluded to my optimal dosage range of 2,000 microliters. I think that if I only did 1 trial, then I wouldn't know the exact range, and if I didn't do 3 trials, then I wouldn't know that the results can vary depending on the culture that they are in and the environment that they're in, and ultimately the dosage of tryptophan administered just depends on a varying of factors, not just, I can't determine that just from doing this experiment alone.
Okay. What are the real world implications? You spoke of that. Can you expand on that? Real world implications or consequences related to your findings?
I think the real world implications can definitely be, this can be applied to humans, except the dosage will be different because humans are obviously different from tetrahymena, and we can't administer dosages as small as microliters. There would have to be something like amount of grams in a capsule. So I think that further research would have to be implemented on furthering organisms that are, I guess, closer to humans. And I just think the overall, this start is a really good starting point for the effects of tryptophan on mood disorders and our overall well-being. And I just think that this isn't enough research to go into administering dosages of tryptophan into humans. So I think this is a good starting base, but the consequences of this, or that it could be harmful to humans, because this doesn't show the whole picture.
Okay, great. One last question. What was the most important research skill you developed as a result of this process, and how might you apply it to your future endeavors?
I definitely think the most important research skill that I did was like doing my own research method and like looking at all of these different organisms under the microscope according to previous studies that I had like looked upon, and like following kind of what they did but in my own way. So like one of the studies that I mentioned was the 2013 research method on tryptophans. Like it was kind of in like a liquid form in like a bottle, like a drink that they would drink, and I think that really helped me form my own research process and my research method, because that showed me like where to start, and ultimately like the processes that they did helped me implement my own solution to an organism. So like I think what I'm trying to say is that the skill that I learned is taking like the real world examples from other people but dialing down on a lower level so I can apply it to organisms.
Okay, all right. Thank you.